Okay, now we're going to look at different assays of tissue response. So, and assays are just ways of assessing the damage from radiation. And so, whatever assay we pick, it should show a clear and quantifiable, that quantifiable just meaning measurable with a number. And also, it should be something that increases. Oops, should be something that increases as the dose is increased. Right. So we already mentioned how chromosomal abnormalities may be one such assay. Um, and so, when we talked about the effects of Hiroshima and Nagasaki, this is what they did, right? They stained and looked at these chromosomal um, you know, mainly the translocations and things. Um, but here we'll also look at other ways that the effects of radiation um, can be quantified. So one of the uh, most common ways is clonogenic assays. So basically, a clonogenic assay is otherwise known as a colony forming assay. It's basically a survival assay based on the ability of a single cell to grow into a colony. So the assay essentially tests um, every cell in the population for its ability to undergo, unlimited division, right? To keep on growing. Um, and clonogenic assay is sort of the method of choice to determine cell reproductive death after treatment with ionizing radiation. Um, but isn't just limited to the effects of radiation. It can be used to determine the effectiveness of other cytotoxic agents, right, such as chemotherapy or whatever. And any substance basically that destroys cells can be measured in this way. Let's look at some of the history of this, though. Uh, many clonogenic assays are in vitro. And when we talk about in vitro, we're sort of talking about in glass, right? Basically, um, while some of them are more in vivo, right? In life. Um, so, for example, rapidly dividing cells um, that are confined to the crypts of the Liber cocoon, they divide about every 12 hours. And this not only replaces their own population, but provides a constant supply of transit cells that ultimately produce the differentiated cells that are, constantly damaged or removed from the villi, right, inside the small intestines, inside our digestive system. Now, if the crypt cells die, then the villi become shortened, flattened, and reduced in number, or even completely absent if the dose is high enough. Um, However, it is assumed that a crypt can be repopulated by just one surviving stem cell. And in these acutely responding normal tissues, survival is usually assayed within a month after radiation because of this rapid turnover of the clonogenic stem cells. And that's basically one way in which um, these can be assayed. So what they did is they looked at crypt survival, um, you know, after irradiation in different parts of a mouse intestine. So mice were either not irradiated at all, the control of the experiment, or they were irradiated with 15 grays of X-rays. Now the images are shown for the proximal, which is the beginning, and distal, or the end, of the small intestine, and at the colon as well. And samples were collected, 3.5 days after the mice were irradiated, so pretty shortly afterwards. The crypt numbers in the irradiated samples tended to be diminished, and they could use this then to quantify the different radiation sensitivities of those cells. And these cells can then be used for, I would say, other cytotoxic agents, and used to investigate potential agents for maybe protecting these cells, right? So there might be something that you could give that might protect the cells from this type of radiation damage. The upshot of all of this is that we can produce these cell survival curves. So on the y-axis, the fraction of surviving crypts is plotted as a function of the dose in grays. One thing to note with all of these survival curves is that we have a log scale on the y-axis. And this sometimes makes it look as if the cells aren't as sensitive to radiation as they really are. And by that what I mean is small changes in the graph. So going down, by maybe halfway down the graph, is actually a much bigger response than it appears, because, in this current graph, for example, going halfway down the graph is going to like 10%. So it's a lot of those cells have been destroyed. Similar assays can be developed for kidney or other late responding normal tissue, and the idea is that the radiation damage to the tissue is the same regardless of how differentiated or slowly dividing the cells are, but that slower dividing cells will express the damage at much later times, maybe over the course of months as opposed to days in the irradiated intestine assays. Another type of assay is transplantation. Transplantation assays are a little strange. Basically, you irradiate or poison an animal and eliminate all of the stem cells in that animal. Then you irradiate a different animal and only partially kill their stem cells. Then you take the cells from the less irradiated animal, which we call the donor, and you put them into the recipient animal, whose cells you just entirely destroyed. For example, mouse bone marrow contains a small population of cells with the capacity to give rise to macroscopic colonies of differentiated cells in the spleen. So here's a survival curve for these colony-forming cells after irradiation with cobalt-60 gamma rays. Both in vitro, in glass, and in vivo, in life, populations are compared, with similar but slightly different results. And actually, the difference is kind of was kind of heavily debated in the paper. They think it might be because the irradiated in vivo cells become more differentiated and lose their capacity to form a colony, or that the in vivo cells are more metabolically active, which means they undergo division more. But the differences were kind of they're not entirely sure of really. But in terms of this assay being able to characterize and quantify the damage done by radiation as a function of dose, it's been very successful. Here's the survival says for a wide range of different cells. So the slopes of the survival curves are similar, and the biggest variation is in the shoulder region at the top of the curves. Now the outlier is the human AT cells, which is a rare inherited disorder that makes these folks more susceptible to developing leukemia and lymphoma, cancer of blood forming and immune system cells, respectively. Another assay would be just the basic lethality, right? So functional assays, especially in late responding tissues, are used to compare tissue responses to radiation based upon the loss of function in the given tissue, right? How the cell is no longer able to do what it's doing, perhaps put it dead. And so after irradiating a specific organ, let's say, for example, 50% of the animals are killed. Right? So in particular, this is represented by the LD50. So this is the dose at which 50% of the animals are likely to die. But also another important measure is the time for this death to occur. And this depends upon the rate of the turnover of the cells, within that given tissue. Here's a series of dose response curves for the lethality of radiation when different normal tissues are subject to localized irradiation. Note that the dose response curves look different from the survival curves. Now we have the response on the y-axis, where increasing dose causes a greater percentage of the animals to die. Okay, and so some tissue and parts of the body are going to have more sensitivity to radiation than others. What's not shown on this curve, though, is the time it takes for the animals to die after being irradiated. So here's a table that shows both the time it takes for the different tissues to die and the LD50, the dose it takes for 50% of the animals to die for different tissues. And so we can see that some parts of the body might be more resistant to radiation than others, but also the time which it presents itself changes too.